Well, welcome back to Bio 180. Today, we're going to continue our discussion of cellular processes. So we're in Unit 5, and we're going to start talking today about the process of cell division. And then we're going to come back in our next lesson, and we'll talk about cell division control. And so that will be next time. And cell division control has implications for cancer, so we'll end up talking a little bit about cancer. But today we're really going to be talking about some of the structures and some of the terms and some of the processes or mechanisms associated with cell division. So that's where we'll start today. So when we come over here, when we talk about cell division, there are really two major processes that cells will go through, at least all eukaryotic cells. Will go through one of two processes. One of them is mitosis, or actually we call it, better said, it's mitotic cell division, and then we often abbreviate it as mitosis. And then the other one is meiotic cell division. And so what's the difference between those two processes? Well, in mitotic cell division, we are going to start with a cell, and we call it a diploid cell. We represent that using the following symbols. We call it a 2N, and that represents what we call a diploid cell. And a diploid cell occurs wherever you have, let's come over here for a second. The diploid number refers to the chromosomes. So we have a cell, and inside that cell we have a nucleus. And inside the nucleus we have our chromosomes. So that's our chromosome. And in a diploid cell, we would have two sets of chromosomes. So this might be chromosome number 1 in red, and this one would come from the maternal parent. We'll call it 1M. And over here we would have the same chromosome number 1, same length and everything. We'll call that 1P coming from the paternal parent. And so what diploid means is that every chromosome is represented twice. That's where the di comes from. So diploidy refers to the number of chromosomes. So this would be a diploid cell. It has one paternal copy of the chromosome and another maternal copy of the chromosome. And then of course you would have different, if this had 4, or maybe we'll make 3 chromosomes, different sizes. And so each chromosome is represented twice, and that's what a diploid cell is. And so now it's clear why we have a 2N up here, right? Because there's 2 copies of each one of those chromosomes, and that would be diploid. And in cell division, or in mitotic cell division, we're going to go from 2N to 4N. We are going to double or duplicate those chromosomes. And so we'll get twice the amount of DNA, and that's why we have now 4N cell. And then we're going to undergo a division process, and we'll go back to having 2 now cells that are 2N. So now in the end of this, we'll create 2 diploid cells. And these diploid cells are exact clones of the original parent cell. And so we use mitotic cell division is used for reproduction, but really only in single-celled organisms. So single-celled organisms use it for reproduction. You and I, as multicellular organisms, we are going to use mitotic cell division for growth. So each one of us starts as a fertilized egg. That's a diploid cell. And that egg divides by mitotic cell division in order to create the 3 billion or trillion, what is it, 65 trillion cells that make up our organism, okay, that make up you. So that's the growth. However, when those cells get damaged, when you get a cut or a scrape or internal organ damage, then it is also used for repair that damage. You'll use mitotic cell division. By far and away, this is the most common cell division that occurs, is mitotic cell division. Now, the contrast to mitotic cell division is meiotic cell division. And so in meiotic cell division, the formula looks a little bit like this. We're going to still start with a diploid cell, so 2N, and then we're still going to double that DNA. We'll talk more about that process in just a moment. And we form the same sort of 4N, because we've doubled it. We do go through a division, but we're going to call this division 1. And we're going to end up with 2 cells that are 2N, kind of similar. And then we undergo a second division. So this is division 2. And what we end up with after division 2 is 4 cells, but each of them are only N. We call this a haploid cell. Haploid means that there's only one representation of each chromosome. So if this were a diploid cell before that we drew, we would simply erase half of those chromosomes. Each chromosome number is still represented, but there's only one copy of each one. So this would be now a haploid cell. So we would now have 4 of those haploid cells. Now haploid cells are not very common in the body. We only find them, the only cells that are haploid in the body are called gamete cells. And the gamete cells are the cells that form, if you're a female, they're what form the eggs. And if you're a male, it's the gametes that form the sperm. So eggs and sperm are types of gamete cells. Up here, the diploid cells that you form, in contrast, the opposite of gamete is a somatic cell. And somatic cell simply means any other cell found in the body. So all the cells in your body are divided into somatic cells. That's what you mostly are. And then everyone has some gamete cells in the reproductive organs, the eggs and sperm. Okay, the gametes are haploid, the somatic cells are diploid. And so meiotic cell division then is really only used to produce gametes for sexual reproduction. Now, I say it's only used for that. That's actually a pretty big deal. And in sexual reproduction, or the reason why we do this is because it allows us to mix and match and create genetic diversity. So we actually, in this process, right here in these first 2 stages, undergo some cellular processes, cellular mechanisms that take those chromosomes and kind of scramble them together, scramble the genetic combination. So that's one scrambling that we get called homologous recombination right here. But then we also get rescrambling because we're going to have eggs from the female get together. And remember, each one of these are haploid, so that's an N cell, and that's an N cell. A sperm from the male and an egg from the female get together, and they fuse together to create a new diploid cell, and this would be the start of a new individual with a different combination. They've got a genetic contribution from the mother and a genetic contribution from the father, but it's all scrambled. It won't be the same as either mother or father was in their diploid cells. So sexual reproduction then allows for a lot of genetic scrambling to occur, and that creates the diversity that we see in the population. Whether it's a room of people or a city of people, we notice that they all look different, and that comes from the genetic diversity that is produced by sexual reproduction. When you're just cloning, if you just use mitosis, you don't get genetic diversity. They're all clones. They're identical copies of each other. That's why all the cells of your body are identical copies of each other. So those are the two types of cell division. And we are going to spend now the rest of our time. Meiotic cell division, this is a Bio 181 topic, and you'll talk about meiotic cell division later on, or meiotic cell division. And then we will continue looking more closely at the mechanisms and vocabulary associated with mitotic cell division today. And this will be the last sort of topic that we cover in Bio 180. So let's come over here. And we often talk about what's called the mitotic division cycle. And so now let's talk about the actual process of cell division and how it works. And so the mitotic cell division is a cycle, meaning it repeats in the same pattern over and over again. And so we'll draw kind of a circle here and go through the different parts of that cycle. So we're going to start right here. About, if this were a clock, we'll start about maybe 10 o'clock. We're going to draw a line, and we're going to divide this circle that represents the cycle into a couple different parts. Roughly, we're going to divide it into about 5 different parts. And we're going to label these. We're going to say this right here is our start point in this cycle. And the start point occurs, this region between this first triangle is called the G1 phase. And what's happening in the G1 phase? Primarily growth. So in this phase, we'll write it up over here. We're going to say in the G1, we're going to get growth. And specifically, we're going to get growth of what we call the cytoplasm. So we just started this cell cycle. It's come off of its previous cell division right here. And so the cell is kind of small, actually. And so it has to grow. It has to essentially double its size. If you had a rough ER, you've got to grow more rough ER. You've got to grow more smooth ER, more Golgi. So you need to grow all of those things, the protein components and everything that's found in the cytoplasm. That's what we're going to do during G1. The next phase is called the S phase. And in the S phase, S stands here for synthesis, which means to create or grow or develop. And what we're doing is we're synthesizing the DNA. So here we'll call this growth of the nucleus. And not necessarily the nucleus itself, but what's inside. So another way of saying this is synthesis. The synthesis phase is where we get DNA replication. So we've talked a little bit about that before in the course. So we're going to get DNA replication. But there's a couple of different ways of saying that. And when we're talking about cell division, you'll see that we talk a lot about chromosomes. We've even already started talking about chromosomes a lot. We said diploid and haploid and sets of chromosomes. And so the chromosomes themselves are made out of DNA. So DNA replication isn't wrong. But you'll often hear us talk about chromosomes being duplicated. So we call this chromosome duplication. as well. That's the exact same thing as DNA replication. Same process, it's just a different way of phrasing it. We would also say that the chromosomes are doubled. And that's what's happening in S phase. We then have another region down here called G2. And in the G2 phase, we are actually continuing. This is with kind of synthesis. This is the edit and repair of chromosomes. And so what we're doing in this stage is, during the replication or synthesis, the cell has to copy somewhere around 3.2 billion base pairs of DNA. So think about that, 3.2 billion. So we represent those base pairs by letters A's, G's, C's, and T's. Imagine if you had to type 3.2 billion characters, an essay 3.2 billion characters long. You're like, I can't even imagine that. We've done some calculations before, and actually if you were to do that and just type it normal standard, single space, 12-point font, double-sided, the number of pages would fill a book that is 70 feet wide. Okay? So that's how much typing that is. And the cell is doing that, and depending on the cell cycle, anywhere from as fast as like an hour to sometimes longer. So it's doing all this work. There's bound to be mistakes, and there are mistakes that are made. And so you need some time, just like when you type out your paper or an essay, you type it out of the rough draft, and then you come back and hopefully you're rereading it and you're correcting all those spelling mistakes, grammar mistakes, word choices, things like that. And so that is really what the G2 phase is. It's kind of this break. We've done most of the typing, if you will, here in the S phase, and then this is just a review and edit part of the cell cycle, and that's called G2. In fact, and this might be as good a time as any to explain, G1 and G2 got their names. The G right here, when I first wrote this up, you might have thought that this stands for growth because that's what's happening. And I don't mind if you think that way. It's not. The G actually stands for gap. And when scientists were first studying the cell cycle, what they noticed is they could find the next stage we're going to talk about, mitosis. It was really easy to see what was happening. The chromosomes would condense and become visible under the microscope, and you could understand and see what was happening in this stage. They could also, using chemical means, be able to tell when synthesis was happening. So it was easy to see this stage, and that there was something big happening in the cell in those 2 points. And then there was this break, or what they called a gap, between mitosis and synthesis. And at that time point, it didn't seem like there was a lot going on in the cell because they couldn't observe anything. And so they called it gap 1. And then the second gap between synthesis and mitosis, they called gap 2. It turns out there's a lot happening in there. We just couldn't observe it directly under the microscope. But that's where they got the name gap 1 and gap 2. And now that we're better cell biologists, we have better techniques. We're not better cell biologists, but we have better techniques. Now we have a better idea of what's going on in those 2 phases. So, following G2, we come to our next stage, which is mitosis. Mitosis. And mitosis specifically refers to the division of the nucleus. Okay? More specifically, the division of the chromosomes. And we'll actually end up going into a lot more detail of what that mitosis is and how it takes place. And then our 5th area up here, this 5th triangle or slice of pie, is called cytoplasmic division. We'll write that up here. Cytoplasmic division. And so this is going to be the division of the cytoplasm. Of all those parts, the mitochondria, the rough ER, smooth ER, all the proteins that are in the cytosol, that's going to be divided during cytoplasmic division. Now, those are basically the 5 phases or steps that we have in the mitotic cell division cycle, but we do have a couple more terms to introduce. These first 3 phases right here, notice that these are all dealing with growth or duplication. So in these first 3 phases, we're growing or we're doubling. If you go back over, remember we went from a diploid cell and we were doubling it to that 4-end cell. So all of these, whether it's doubling of the cytoplasm or doubling of the chromosomes, these are all associated with doubling. And we call this interphase. All of those together, we'll put this is the growth phase, if you will, to summarize what happens there, occurs in interphase. This by far is the longer part of the cell cycle. It can last anywhere from about, usually it represents about 3/4, sometimes as much as, 23/24. A typical cell right here might spend, out of a 24-hour cycle, it would spend 23 hours here in interphase. And then the last hour will be spent in this next part, which is called M phase. So M phase, or the mitotic phase, combines both mitosis and cytoplasm. You might have been wondering why we didn't call this one the M phase. It's called mitosis because the M phase actually includes both of these put together. Okay? So that's how we divide it up and some of the vocabulary associated with the different phases. Now, let's talk about one more thing before we start to get into our mechanisms. And let's talk about the chromosomes. Chromosome terms. And let's explain a couple different chromosome terms. Because you might imagine, since we're talking about both the creation or doubling of the chromosomes and the splitting of the chromosomes, that the word chromosome gets thrown around a lot in those types of conversations. And so, a couple of terms. One of the things that you'll hear is a term called the genome. Okay? What is a genome? This is just a sum total of cellular DNA. All of the DNA inside the cell. That includes the nuclear DNA, all the different chromosomes. It actually also includes things like DNA found in the mitochondria. But you often hear it's just a shortcut where we'll say we're going to divide the genome, or the human genome has so many chromosomes would be a common way of expressing that. That means the cellular DNA is divided up into 46 chromosomes. Okay? So that's what genome means. You'll hear the term, obviously, chromosome. So let's talk about that for a second. And what is a chromosome? A chromosome is a single continuous double helix of DNA. Okay? So that would be a single chromosome. Usually this refers to just being the DNA is what we're mostly focusing on. You'll hear another term called chromatin. Now, chromatin is a little bit different than the word chromosome, but they're often used interchangeably. Chromatin is equal to DNA plus proteins like histones. So DNA itself, when it's found inside the cell, does not exist in a naked state. It's not just raw DNA. That DNA is bound by a whole host of different proteins. And one of the most common is a protein called a histone, a DNA binding protein that acts as a spool, so DNA kind of winds around all these different histones. It's a way of packing the DNA. But it's a very common protein found in the DNA. And so when we talk about chromatin, we're talking about a complex, a mixture of both DNA and proteins, kind of in its native state inside of the cell. Notice I didn't put single continuous strands or what. Chromatin usually is referring to all the DNA, if you will. And it doesn't specify individual chromosomes within it. And so a lot of times, so this would be definition 1, kind of an unofficial definition of chromatin when we're talking about that, is it refers to DNA in a uncondensed state. And then a lot of times up here with this word chromosome, when you hear people talking about the chromosomes, a lot of times when they draw a picture of a chromosome, they're drawing it, the DNA, in its condensed state. So both of these words kind of have a second meaning of being condensed or uncondensed. And let's talk about that for a second on what it means to be condensed or uncondensed. If we have a nucleus, and an uncondensed chromosome would kind of be long and not very structured, so this might be an uncondensed chromosome. And then when it condenses, it kind of loops together and weaves together, and it would form more like this fat finger type structure that you're often more used to thinking of when you think of a chromosome. So this, both of these are chromosomes, but this would be a condensed chromosome. And this one would be an uncondensed chromosome. Okay, so this one will be all spread out. The uncondensed form is usually the functional form. That's the form that the DNA is in most of the time. So when the cell is in interphase, all through interphase, we're in an uncondensed form. And then we only condense the chromosomes themselves when we're getting ready to move them, when we're actually dividing them during mitosis. Why do we do that? Well, you might imagine you guys face the same problem. The end of the semester is coming up soon, and you're probably, many of you, getting ready to go home. Well, you've got all your stuff spread about your apartment, all your clothes and your toothpaste and whatever else you have. And it's spread across your apartment in its functional form, right? But it's not very good for traveling. Could you imagine maybe going out to the airport and you just kind of grab a big armful of clothes and you walk into the airport and you set them down on the counter, you know, I need one ticket to Des Moines. And you're moving through. As you do that, if you haven't packed those clothes and all your things together, you're going to be leaving pieces and parts of you all over the place. Dropping them here, it's going to be lost, it's going to be damaged. And the same thing happens with our chromosomes. The chromosomes themselves need to be open and spread out to be functional. You have to have gene or proteins that can come in and copy them and make RNA and all that. And so you have to be open and functional and uncondensed most of the time. That's how you operate them. But in cell division, we're going to move them to opposite sides of the cell, or to the nucleus or the cell. So if I grab this one and it was spread out and I pull it to the other side of the cell, there could still be pieces hanging over from where I originally pulled it to. And so it wouldn't move very well. So what the cell does is it takes those long strands and it compacts them into a nice, tight structure like this that's visible actually under a microscope. And now if I grab this part and move it, the whole thing comes with me. It's like the difference between grabbing an electrical cord and dragging it wherever I need it, or coiling it up first and then pulling it to where I need it. And that's what's happening. So we are going to condense the chromosomes. Now, you are probably, although I've drawn the condensed chromosome in this form, you're probably more used to seeing a chromosome in this X-like structure, right? Maybe from high school or biology. Both of these would be representations of condensed chromosomes. The difference is another feature of chromosomes. This is called a duplicated chromosome, okay? And a duplicated chromosome, both of them are one chromosome. This would be unduplicated. And this one will be duplicated. Both of those, though, are one single chromosome. We do run into a little bit of ambiguity, though, in that we look at them, and if we call both of them a chromosome, you're kind of looking at that saying, yeah, but one of them is different than the other one. You know, one of them seems to have more stuff. Well, it does, because it's been duplicated. However, we do want to give a name to the more stuff. So we would say that this duplicated chromosome is made out of two parts. And the parts, we would say this would be half of it that I circled in blue. And that part is called a chromatid. So in blue, I've circled one chromatid. And then the other chromatid would be right here. So a duplicated chromosome has two chromatids. In fact, let's write that as a definition. We're going to say that the word chromatid is going to equal 1/2 of a duplicated chromosome. And we have to be careful about that definition, duplicated chromosome. So right here we have a duplicated chromosome, and it would have two chromatids. This would be one chromatid, and that would be two chromatids. But when we come up here to our unduplicated chromosome, and this is causes a lot of consternation, a lot of problems for beginning biology students studying genetics and cell division. When we look at this chromosome right here, and we ask ourselves, okay, how many chromatids does this chromosome have? And the answer, I see some of you got 0, some 1, no one said 2, that's good. The answer is that this one has 0 chromatids. Well, why does it have 0 chromatids? Because a chromatid is 1/2 of a duplicated chromosome. So you have to ask yourself, how many duplicated chromosomes do I have? Well, none, or 0. Well, what's 1/2 of 0? Well, 0. So this one doesn't, in other words, in the unduplicated state, there's no need to have a term like chromatid. There's no ambiguity. But here, we do, in a duplicated chromosome, often need to be able to talk about what the two parts are and call them chromatids. So that's going to come up, and we use that term quite frequently, and that's the explanation of it. So our chromosomes, and we're going to see during the cell cycle, have different states that they're in. They could be condensed or uncondensed, and they can be duplicated or unduplicated. It's sort of like a person could have blonde hair and blue eyes, or they could have brown hair or green eyes, and then you get various combinations, right? There are two different features, whether you're duplicated or not, or whether you're condensed or not. And knowing what those are and the states that they're in is important. So let's go through and practice using some of these terms by looking at the mitotic cell division cycle. And let's describe the state of the chromosomes in each of these units. Okay? Now, for this, we're going to use a human cell. And the diploid number, 2n, equals 46. So that means that the chromosome in a somatic cell like we're looking at would have 46 chromosomes. 23 of them would come from mom, 23 of them would come from dad. And that would be the two sets of chromosomes. So when we look at G1, is the chromosomes in G1 going to be condensed or uncondensed? Think about that for a second. In G1, we're going to be uncondensed. Okay? And the next question is going to be, are we going to be duplicated or unduplicated? Well, in G1, we haven't done our chromosome duplication yet, right? That occurs in S. So in G1, we're unduplicated. We might also ask ourselves, this would be a good test question, how many chromatids do we have in G1? And the answer is 0. So we'll put that up. Chromatids equals 0. How many chromosomes do we have? Well, we have 46. Okay? Now, maybe we'll write that up there too, in purple. Chromosomes equals 46. Now, let's move on to S phase. In S phase, what are we? Well, we are still going to be uncondensed. Uncondensed. Now, up here, we were unduplicated, but this is where duplication occurs. So, depending on where you are in the S phase, we're going to put that here. They are duplicate. And instead of duplicated, meaning it's done, well, I'm going to put down duplicating. So it gets a little bit complex in S phase because they're in the process of being duplicated. The number of chromosomes in purple is still 46. The number of chromatids well, it varies. It's sort of undefined in S phase because we're in the process of duplicating them, and so you're in the process of creating the chromatids that are in a duplicated chromosome. So we're going to leave it as undefined or varied in S phase. But we do start to be able to define it once we get here to G2. So to kind of speed this up, in G2 we're still uncondensed. However, we've gone through S phase, so we now have duplicated chromosomes. We're now fully duplicated. The number of chromosomes is still 46. So we actually have the same number of chromosomes as we did when we started the process. But the difference is those chromosomes are now all duplicated. So our chromatids, how many chromatids do we have? Well, for every chromosome, duplicated chromosome, we have 2. So that means we have 92 chromatids at that point. Now, in mitosis, we finally get some variation in the blue. So now, this is mitosis, pretty early in the process. We are condensing our chromosomes. So this is the one area where we are now condensed in our chromosomes because mitosis is where we're going to be moving those chromosomes. So we had to pack. So they're condensed. They are, we'll put duplicated. At least they start out that way. And then I'm going to put a question mark in here. Remember, during mitosis, this is the point where we divide them. So they're going to go from duplicated to, again, being unduplicated. So just like it was kind of undefined over here, the start of it, they're duplicated. By the end of it, they're not duplicated. Here, chromosomes, well, this gets a little bit funny. We're going to start out with 46 duplicated chromosomes. In a process called anaphase, we're really going to start to separate those, and then there'll be individual chromosomes. So kind of before we split the cell, we're going to go anywhere from 46 to sort of having 92 at some point, at least for a brief point, right? And we'll show you where that occurs. And then chromatids gets a little bit funny too. We know that we start out with 92 because we're coming out of G2. And then as we split them, we often, in fact, in an anaphase, we often refer to it as we say we're splitting the sister chromatids, the two connected chromatids, we're splitting them. And of course, when we split them, then they're not chromatids anymore. So eventually, we're kind of going toward the direction of having 0 again. But a lot of times we would say in mitosis, we have 92 chromatids present, at least at the start of it. Okay? And then 46 chromosomes, at least at the start of it. And that's sort of how the DNA changes throughout that cell cycle. Now, so this guy's good. We've learned a lot of the different terms associated with the mitotic cell division cycle and with chromosomes. We're going to take this area right here, mitosis, because we could see it first in the microscope. And there's a lot going on. We happen to know a lot about what is going on in this process. And so we're going to take a closer look at the structures involved and the process of dividing those chromosomes and separating them. While I'm over here, let's go ahead and for a moment talk about S phase. There's a lot. Remember we said we knew a lot about this too. So why aren't we talking about that? Because again, this is going to be a Bio 181 topic. So when you go into Bio 180, you're going to spend almost probably a full week just talking about RNA or DNA replication and what happens and how we do it. So you'll cover this in Bio 181 next semester, and then we're going to focus on these events of mitosis right now. Mitosis, it turns out, there's a lot happening in mitosis. And we actually, if we represent that, remember as this part of the pie, we actually have divided into 5 separate events or phases. And that's what we're going to talk about and a little bit about what's happening in each one of those phases. So we have right here prophase. They all end with phase. So we have prophase. We have prometaphase. We have metaphase, anaphase, and telophase. And we'll talk about what's happening in each one of those. So we'll start with prophase. Okay? And when the cell first enters into mitosis, prophase, what's happening there? Well, let's maybe come over here and briefly draw a picture of the cell, maybe at the end of G2. And then we'll try and write over here the different things that are happening, and then illustrate over here what's happening inside of the cell. So let's draw a cell. And we're going to say at the end of G2, what does that cell look like? It's got a plasma membrane. Here in the middle, nucleus. And then inside of that nucleus, we have got our chromosomes. However, they're uncondensed. They are duplicated, but they're uncondensed. And so they kind of look like, we'll duplicate them. They're kind of long and spread out here. And then we've got another one. And we might have, we're not going to draw all 46 of them, but they're spread out. They're kind of chaotic in here. We'll put another little one right here. And hopefully you guys are starting to get the picture here. Of what it looks like. So we've got I don't know, maybe 6 or 7, 8 different chromosomes in here. This is what we'd often refer to. They look at that, and you can't pick out individual chromosomes. So cell biologists would just call this a big mass of chromatin. We can't pick out chromosomes. It's in its chromatin state, we might say, or uncondensed state. And that's what it looks like. We have another structure. We've learned about this previously over here called the centrosome. Okay? And the centrosome, you might remember from our previous lesson on the cytoskeleton that the centrosome is the organizing center of the microtubules. I'm just going to abbreviate microtubules as MT. We'll be using that a lot. Now, one of the things that has happened during G2 is we've taken the centrosome and we've duplicated it. And so we now have, just like we duplicated, we now have actually 2 centrosomes in there. And that becomes important in a second. So this is at the end of G2. So what happens in prophase? Well, one of the first things that happens is that the chromosomes condense. Okay? So the chromosomes are going to condense. There's also, this is not a big one, I don't really care, but you'll see this in a lot of textbooks. As the chromosome condense, you remember when we talked about the parts of the cell earlier, there was a part inside the nucleus called the nucleolus. The nucleolus was kind of just a darker spot that you could kind of observe inside the nucleus. And so this was the nucleolus. You'd see that. It's really combined. It's just a dark spot because you get a high concentration of chromatin in that area due to the making of ribosomes. And so when you condense the chromosomes, then that high concentration gets pulled away, if you will. And so they say that the nucleolus disappears. And that's just what they observe. The nucleolus is disappearing is because the chromosomes are condensing. And that's generally what we talk about with prophase as what's happening. The next part, we also started, we'll put under here the nuclear envelope. Breaks down. Okay? And then we go on to our next stage, which is Prometaphase. In prophase, for our Prometaphase, one of the key things that we're going to do is get formation. In fact, I'm going to switch these. We'll start this up here. You get formation of the spindle assembly. And then we'll put down nuclear envelope breaks down. And so I kind of reversed the way I'd had these written. And that's, it matters, but it doesn't matter a lot in the sense that both of these processes are starting in prophase, at least initially, and then they're going to end in prometaphase. And they're kind of crossing over this boundary a little bit and occurring a little bit in both stages. Now, let me explain over here what we mean by this term spindle assembly, because this is a big deal. The spindle assembly has to do right here with our centrosomes and the microtubules that come out of them. And what happens is that we have microtubules that are going to grow out from this side, and microtubules that'll grow out from that side. The way this works, we talked about plus and minus ends on microtubules. The plus ends are right here on the end of it, and then over here, let's draw a couple more. Plus end here, plus end there. And what happens is there is a motor protein right here. It's a kinesin. Kinesin motor protein. And the kinesin motor protein is actually connecting those two opposing microtubules. Remember, kinesin motor proteins walk or move in the plus direction. And this particular kinesin protein has a motor on both of the different filaments. And so what happens is if my hand is kind of the kinesin motor, I'm the kinesin sort of shoulder joints, and I have one hand on one of the spindles sort of right here, and the other hand on this one. And so it has both of them. And as it moves toward the plus direction on each of those spindles, it actually pushes them apart. So I grab plus end over here and plus end over here, and then I move them apart, and then it ends up as the microtubules keep growing, and I keep grabbing and walking along them, it causes whatever's connected to the other end of those microtubules, meaning the two poles. We call these often the poles, or the centromeres, or sorry, the centrosomes. We often call them the poles. It pushes those two poles apart. And I realize that could be a little bit confusing. By pole, we mean like North Pole and South Pole, okay, not like a rod. So it pushes the two poles apart. And so this is what we mean by the spindle assembly. So that by the time we get to the end of prophase, let me show you what we mean a little bit by this. So prophase then starts to look a little bit like this, in that we now have our chromosomes are now condensed. Into discrete structures. They're still all kind of jumbled around in there. But we now have condensed chromosomes. That happened early on. The nucleolus has disappeared. Our spindle apparatus, or assembly, starts to look then a little bit like this. I'm going to take those centrosomes, instead of drawing the sort of tin can perpendicular structure, I'm just going to use a purple dot. So that's the centrosome. And we now have, they're starting to separate. They do have other microtubules going in other directions. And we have that kinesin motor protein in between them that's causing them to keep pushing apart, right? And so this is, by the end of prophase, this is kind of what it looks like. We're starting to form the spindle assembly. This is eventually it's going to keep dividing, and then at some point it's also going to shift itself. I couldn't draw it across, so it's going to shift itself and separate to these two sides, okay? The nuclear envelope, it still looks pretty solid, but it's going to start getting some gaps in it, okay? So that's about what we're looking at by the end of prophase. Now, in prometaphase, we said the nuclear envelope breaks down. We get full formation of the spindle assembly. So the spindle fully forms. And we also, toward the end of prometaphase, we're going to get something called the kinetochore microtubules contact kinetochores. Now, we got some new terms in there. The kinetochore microtubules is one term, and then we have the kinetochores is the other term. So let's update our illustration now to no longer be prophase, but now we're going to draw prometaphase and see what happens in prometaphase. So one of the first things that happen in prometaphase is that we get the complete dissolution of the nuclear envelope. So we're going to completely erase that. What's really happening, I just kind of erased it. You might say, well, what's really happening in the cell? The nuclear envelope, we call it vesicularizing. It forms little vesicles, and those vesicles are going to spread all over. Current understanding is that the little vesicles of the nuclear membrane actually hitch rides on the chromosome arms. But that's a whole different story. So we're going to erase these, get rid of this. But it's basically forming little small vesicles that we can't see under the microscope. We're going to redraw our spindle assembly. It's now fully mature. We've got our poles on different sides of the cell. So that's our centrosome structure. We have microtubules still coming out from them. So we've got these long microtubules now. They've really been growing on us. And let's start to identify, let's draw our kinesin. We've got our kinesin motor protein right here. And it's still pulling and pushing. Kinesin, forming, keeping those spindles apart. Now, right here, these structures, we have microtubules coming out of the centrosomes on either side, on either pole. We start to be able to name them at this point. They're all microtubules. They're all made out of tubulin. But depending on what they're attached to, they have different names. So these microtubules that are kinesin and they're pushing apart each other, these are called overlap microtubules, or sometimes you hear them called non-kinetochore microtubules. These ones down here at the bottom are actually connecting and anchoring the centrosome to the plasma membrane. And you'll see there's a lot of around here. There'll be a lot of little short ones that kind of come out like this, kind of non-specific. And these ones are called aster microtubules. And then you'll see some others that'll emanate from, and they go directly to and contact the chromosomes. And these ones are then called kinetochore microtubules. Kinetochore microtubules. Now, to understand why they're called kinetochore microtubules, let's come back over here and talk about chromosome structure for just a moment. So let's draw a duplicated chromosome. And this region right here, these kind of bulbs, this is called the arms of the chromosome. So that region is called the arms, and all the arms of the chromosome are connected at this pinch point in the middle, and that is called a centromere. And that's the pinch point, so we'll put right here the pinch point. And then at the centromere, on either side, they have two of them. 180 degrees from each other, you have this special collection of proteins that is known as the kinetochore. And the kinetochore forms the docking site for that centromere. What is the docking site for what? The docking site for the kinetochore microtubules. So the kinetochore microtubules are going to come and land and dock and attach to the centromere through those kinetochores, the proteins associated with those kinetochores. There is one more significant thing. These kinetochores also have a dyning motor inside of them. That's one of the proteins contained in the kinetochores is a dyning motor. And that's going to come up in a second. So just remember we have a dyning motor in there. So what we mean by this is actually the centrosomes from either side are sending out these kinetochore microtubules to contact the different chromosomes. I probably do too many chromosomes here. But every chromosome ends up getting contacted by a kinetochore from one side and then a kinetochore from the other side. We're not going to go into a whole lot of detail about it, but what actually happens is kinetochores are kind of randomly, the kinetochore microtubules will grow and shrink, and they're kind of flung out randomly, almost like a fly fisherman casting and then reeling back in. And the kinetochore, or sorry, the centrosome right here is just kind of shooting out. Remember, they're dynamic, so it's shooting out these microtubules until one of them hits a kinetochore. And when it finds a kinetochore, it grabs onto it, and then it actually starts reeling it in. And so the kinetochore will start to move this way. From the other side, you have random shootings as well. And so you eventually get to a point where the kinetochores will get contacted from each side, and then you start this tug of war that starts to happen between the two poles and their associated microtubules. They pull this way and they pull that way. There's also present here, you don't have to really worry about this too much, but there's also kinesins on the arms that will contact overlaps and pull the chromosomes this way. So there's a lot of pushing and pulling going in on those chromosomes. The end result of all this pushing, and you can actually watch this under the microscope, you can see these chromosomes, they'll move one way, and then they'll start to move the other way and go across the cell, and then they'll start to move the other way. And they kind of go back and forth until they end up right in the middle of the cell. And that's where they're supposed to be. All this pushing and pulling and balancing of forces leads to those chromosomes lining up right along the middle of the cell. And that is what signifies metaphase. So metaphase is characterized by the chromosomes lining up along the middle of the cell. Chromosomes align in the middle of the cell. We call this the metaphase plate, and that's what I'm going to write here. Align on the metaphase plate. And that simply means the middle of the cell. Okay, so they align along the metaphase plate, and that characterizes metaphase. And then they just sit there for a while. We'll explain why they sit there for a while in our next lesson when we talk about cancer. So what does this look like? I'm not going to draw all of them that I had before. We're just going to draw 3 of them here. So each one is still being contacted by their kinetochore microtubule. But they're all aligned right along the metaphase plate now. And so this would actually be then metaphase. And this is what you look at. And it's really easy to identify. This is what you often see cells when you're looking under a microscope at rapidly dividing cells. You'll be easily able to identify a cell in metaphase because you'll see the chromosomes are all just lined up right along the middle. And then they kind of, right here, they pause. So they align, and then they kind of just sit there and pause, and they're waiting for a signal from the cell. We'll see later that this is a cell division checkpoint right at the middle of metaphase. In fact, I'll write that, pause in parentheses. We'll put checkpoint. And then we'll come back and explain that in our next lecture. What happens after metaphase? Well, we have anaphase. And anaphase is where we split and separate sister chromatids. So here's that word chromatids again. And then we also, in anaphase, we somewhat lengthen the cell. So we widen the cell as well. So what happens in anaphase? Right here, there's a protein involved called cohesin that sort of stitches these two sister chromatids together and is holding them together. It's located in the centromere and actually all along both arms. And that's what's keeping them. You're pulling from each kinetochore, but they can't separate because they're all stitched together with this cohesin protein. And what happens is, so you got them all stitched together. At the beginning of anaphase, there is a protein called, and actually this will make sense, its name is Separase. And so you have cohesin right here, and then it gets eaten. Cohesin is eaten or digested or broken down by Separase. And so Separase comes along and it just breaks all the stitches. Now there's nothing holding those sister chromatids together, but you do have the two kinetochores now. They've been released. They were being stitched together, but they're not anymore. And remember that at each kinetochore, you had that dyning motor protein. Well, remember, dyning motor proteins, kinesins walk toward the plus end, dynings walk toward the minus end. And so the dyning, where's the minus end? Right back here at the poles, at the centrosomes. So that means that this dyning starts pulling the chromosome that, or the chromatid that it's attached to in this direction. And so you get movement of the separation of the chromatids or chromosomes in that direction. So what happens is you no longer have this in anaphase. They've now been split, and so they start to separate to opposite poles. Now, I haven't quite drawn things to scale here, in that inside the cell, the chromosomes are actually much larger than what they're appearing. And this is the reason why you still need these overlap microtubules. If we didn't have these here, and the pole tried to move the chromosome mass toward it, it actually would push the pole toward the chromosomes because it's much bigger. It's like me trying to push, I don't know, like, this desk. If it's really heavy and I try and pull it toward me, I could just pull me toward it. But if I push against something, like I'm pushing against the microtubules with one hand, like the overlap, and now I try pulling, now I'm pushing and pulling those chromosomes, and they'll move. Okay? And so that's what's happening. We had to have these overlaps to kind of be pushing against and then pull the chromosomes to the opposite sides. And that is anaphase. there's also the overlaps keep separating, and so the cell will actually lengthen a little bit as well. It's going to kind of grow a little bit outward as well as it's pulling the chromosomes. And then we have our last phase, which is going to be telophase. Telophase is kind of a reversal of prophase and prometaphase. I'm running out of room right here. Oops, I guess we need to rewrite this as anaphase is what we were drawing. And so now, I'm going to rewrite it again. We've got our last one, telophase. So what happens in telophase? In telophase, we now have our chromosomes, and they have been separated to the opposite sides of the cell. And now what happens is the nucleus, remember we said that the little nuclear vesicles were hitching rides on the chromosome arms? Well, they were. And now what happens is they all start to come together and they start to refuse back to each other. And so you start to reform the nuclear envelope. And so on each side, as those vesicles all come together and start fusing, that's why I'm drawing this as a bunch of little circles, they start fusing. And so the first thing that happens is you get reformation of the nucleus. Then inside, the second thing, big thing that's happening in telophase is that inside of these, the chromosomes are then uncondensing again. And we'll get the reformation, if you want to put it in there, of the nucleolus on each side. And so now, for a brief moment, we actually have two nuclei inside of the cell. However, it doesn't last for long. Because the second thing, mitosis was the division of the nucleus. We've now divided the nucleus. We now have a separate process called cytokinesis, or cytoplasmic division. In fact, I think I called it cytoplasmic division earlier, but we're going to give you the more common name, cytokinesis. So cytokinesis. And this is not actually part of mitosis. This is a separate process. And remember, this is the splitting of the cytosol. And what happens in this process is we are going to form what we call a contractile bundle of actin and myosin at the metaphase plate. And then we're going to get contraction. Remember, actin and myosin, these are the two components. Actin is the cytoskeleton protein. Myosin is the motor protein. These are the same two proteins that make up muscle. And in fact, what we're going to do is right along here, so they form at the midplate, sort of like this. So here's the actin filaments that form. And then you get this myosin motor protein, and it starts to act just like a muscle. The myosin starts to contract and move these together, and it forms this contraction, this ring. So we'll call it the contractile bundle. We also sometimes call it the ring, contractile ring. that ring starts to pinch together. It's attached to the plasma membrane on the outside. There's proteins that attach it to the actin filaments. And so what starts to happen, we're no longer in telophase. We're now in cytokinesis, is we start to get the plasma membrane pulling in at the midplate. It collapses, so to speak. And this indentation is referred to as the cleavage furrow. We have the cleavage furrow. And that contractile ring continues to press upon those and bring them together until you get this really narrow band right there that's still holding the cells together. And then some other protein, we're not sure about this process, but it pinches them off and creates the two new cells. And so that's cytoplasmic division, or cytokinesis. And we're separating the cytoplasm of those two cells. So right here, the contractile ring contracts, forming a cleavage furrow on the plasma membrane. And that eventually separates the two cells. Now, we call these two different processes, cytokinesis and mitosis. They can actually be separated. So in the development stages of certain organisms, you might get cytokinesis happening, meaning the cell will divide, or actually not, this doesn't happen naturally, but you can actually trick the cell into dividing, splitting the cytoplasm, going through the pinching process without separating its chromosomes. So you can trick the cell, it'll split, even though it will only have one nucleus. So one cell remains, or becomes anucleate. There's no nucleus in it. So you can get the cells to pinch without dividing. You can also, this does happen, you can get mitosis happening. You can get the nuclei dividing, but not cytokinesis. They won't split. They'll just have multinucleate in certain beginning stages of development. So cytokinesis and mitosis are actually two different processes. Although they're pretty connected, they're both dividing, and they happen a lot of times together. Cytokinesis will often actually, this contractile ring will often start forming in late anaphase, and you'll get contraction during telophase. So telophase and cytokinesis will often happen at about the same time. One more thing that we want to talk about, and then we'll be done with our mechanisms. And that is, let's erase this. And let's talk for a second about plant cells, because they face kind of a unique problem in that a plant cell, to do cytokinesis, so we'll write up here cytokinesis in plants. And the problem with plant cells is that, remember, they have this rigid cell wall that surrounds the whole plant. And that rigid cell wall will keep the cell from forming a cleavage furrow. So right here we have the cell wall. And so we can't get a cleavage furrow because it can't bend in or bring in that rigid cell wall. So what do plants do? How do you do cytokinesis in plants? Oh, we'll form, let's draw our two nuclei over here. So that's our nucleus. To do this, we need another organelle, and that is, see if you guys can recognize this one. We have multiple compartments. We need our Golgi. So the Golgi starts to reform. And what happens is the Golgi will actually send out vesicles. And those vesicles are going to be filled with cell wall components. So I'm going to put a little CW in each one. And they're the components, the polysaccharides and proteins that actually make up the cell wall. And so the Golgi will excrete these, and then these components sort of end up lining along the midplate. And they have their cell wall components. And then these components start to fuse, these vesicles all start to fuse together. And they actually then start to make, remember that they have those cell wall components. And so in situ, they sort of build a new cell wall. And then all the vesicle membranes actually fuse into the new plasma membranes. And then we have two new cells. And so plants approach this differently. Rather than pinching their cells into two different parts, they simply build a wall between the two, okay? And that's how plants do it. And then these cells would enter into another G1 and start to grow out and elongate and become a little bit bigger. So that's how we perform cytokinesis in plants. So just to summarize a few things, when we started out this lesson, we were talking about the different types of cell division, meiotic and mitotic cell division. Then we focused on the mitotic division cycle. We went through the different major phases of that cycle and talked about what's happening in each one. We talked about some of the chromosome terms like duplicated, unduplicated, condensed, uncondensed, sister chromatids. And then we went and started to focus really on the structures and mechanisms associated with mitosis and cytokinesis right here. And then we talked about cytokinesis in both animals and plant cells. And that'll wrap it up for this lesson. Good luck.